Welcome back, everyone. Please give me a thumbs up if I'm audible and visible. Okay, wonderful. So let's begin. There was a slight delay because there were some changes that we had to make in the PDF that you have been given to you. So if you have downloaded the previous PDF, just make sure that you download the new one as well, because the new PDF is little different from the older PDF by that we by mistake had uploaded. So that's why it took us some extra minutes to come back. Sorry for the delay. All right. So, moving on from where we were, so we had discussed mainly about what exactly a transgenic crop is, where we are saying that there is a gene which is coming from some other organism, that there's some other organism where we'll find some gene which is useful for us, and this gene will be inserted inside this given plant. Now, how is this process basically, and what is the entire procedure? Now, is it something very simple that we'll just go find that gene, cut it out, replace it with another gene, and we are done? Unfortunately, things are not this simple. And the entire technology, the way the entire technology works, could be a little difficult to implement. And that's why genetic engineering, many a times we see that is not even very stable. So that's why we see that there are certain steps. Now, when it comes to this technology where we replace the gene or we are modifying the gene, the technology is called as recombinant DNA technology. And with the help of recombinant DNA technology, we make the changes in the plant. Now, how exactly this recombinant DNA technology works, let's try to understand that. So what we discussed in some of the examples, for example, we saw BT cotton and we saw golden rice, correct? We saw these two examples. Now, what we know about in these two examples is that in all these two cases, there is an external gene that we are inserting. All right. There is an external gene that we are inserting, and this external gene has that quality that we are looking for. For example, in case of BT cotton, we took a bacterial gene, and this bacterial gene could help us in fighting the pest, or it could help the plant in becoming poisonous for those pests. So that was the gene of interest for us, right? That was the gene of interest. Similarly, in case of BT cotton, what we derive from beta carotene and how beta carotene helps in the synthesis of vitamin A, this beta carotene comes from a gene, and that's why that is the gene with beta carotene is the gene of interest for us. So first and foremost, the first step to any such genetic engineering or any genetic modification is the identification of the gene. So first and foremost, we will identify the gene of interest, correct? We'll identify the gene of interest, and once we have identified it, this is where we move on to the next step. Now this is where, as I told you, that it's not that you'll just take the gene and insert it wherever you want. First, we will have to find a carrier. All right, there has to be a carrier that will carry this gene inside the plant. Now, what is this carrier? So this carrier, also called as a vector, would be a virus or even could be a bacteria. All right. So in this case, when it comes to the gene of interest, we need a carrier, or what we also call as a vector. And this carrier of the vector can be a virus or bacteria. So we can choose a virus or we can choose bacteria, and that basically becomes the gene of interest. So, sorry, that will become the carrier for the gene of interest. So overall, we'll find this carrier. Now, in this carrier, this gene has to be inserted. Now, inserting that gene into a given location is also not a very easy task, because here also we see that you have to find a matching sequence. Just think of it as a jigsaw puzzle, that in a jigsaw puzzle there are missing pieces. So it's not that you can simply put any piece wherever you want. Every piece will have its own place. So unless you find that exact piece that can set in that particular jigsaw puzzle, you can't really solve it. Same happens here as well, that for the gene of interest that we have chosen, we have to find a vector and a place in that vector, in the genetic structure of that vector, where you'll be able to insert this gene of interest. So that is the second thing that we do, that we find the vector and we find the required position in the vector, in that vector, so that we can insert this gene. So for example, if you see this, we would make a cut something like this. All right, we will make a cut something like this, because when we cut open this, we will be able to insert this gene. So just look at this, that what will happen, that how exactly does it look like, that this vector, when it has been cut open, we'll see that this will look something like this, that it has been cut open. All right, so we are actually leaving this space. So this space that has been left, this will be occupied, this will be occupied by this gene of interest. So this gene of interest basically will occupy this space. All right, this gene of interest will occupy this particular space, correct? So that's why matching sequences are very, very important, that we'll have to find an area, or we'll have to find a place in the genetic sequence of the carrier where you can insert this gene. So once we have inserted this gene, the next thing is to close the ends or make the ends to stick together. So we apply a Fevi-stick or a Fevicol or a Feviquick here, but of course a normal Feviquick will not be able to do anything. Here we have special enzymes that are used so that we can make the ends to stick together. All right, so these enzymes are basically called as uh, DNA ligase. All right, so this DNA ligase will act as an enzyme that will make the ends to stick together. So that's why there are all these enzymes that help. For example, even in cutting the gene at the given required location, there is a particular enzyme called as the restriction enzyme. So the role of the restriction enzyme is to go to the place, find the place where the cut has to be made. So that's why it will find the way, it will find the place where the cut has to be made, and it will make the cut in the vector. And once the vector has been cut open, we insert the gene of request in this vector, and then we use DNA ligase. Then we use DNA ligase, and with the help of DNA ligase, we'll be able to, with the help of DNA ligase, we'll be able to make the ends to stick together. So now we have a vector which has the required DNA sequence. Now this, what the next step also still is not insertion into the plant, but before that there is another process that will be carried on, and this process is PCR, polymerase chain reaction. Now this PCR basically is where we are creating multiple copies of this DNA or of this genetic sequence, multiple copies. It's just that, just like somebody has made the notes and there are thousands of students who are just photocopying the notes. So similarly, there are thousands to lakhs of copies that are created for this recombinant DNA. This DNA which has the gene of interest, now it is called as the recombinant DNA. So this recombinant DNA, uh, now that we have, basically there are multiple copies that will make of it, and then finally it is inserted inside the plant. So that's why unfortunately it's not an easy process that you just cut the gene and just insert whatever you do. There was an advertisement for Godrej hair dye, one used to come once, काटो खोलो घोलो और लगा लो और हर सफेद बाल काला बना लो, they used to say. So unfortunately things are not that simple here, that we have to actually go through this entire process so that we can set up this particular segment in the required location, and then this DNA, it will work along with the given plant, and hence it will be able to now add to the value of this plant by having this required gene, the gene of interest here in this particular plant. So this is basically how we go, this is what we do, and this is called as recombinant DNA technology. So if you just let me just go through the steps once more for you. So first and foremost, the first step is to isolate the desired DNA. That we first have to identify the DNA, and we have to isolate the DNA in its pure form. All right, it has to be identified in its pure form. So that's the first step that we have here. The next step for us is to cut the gene at recognition sites. Cutting the genes at recognition site, which will be done with the help of restriction enzymes. As I told you, that restriction enzymes will find the place where the cut has to be made. They'll find the place where the cut has to be made, and it will go there and it will make the required cut. All right, so it will go there and it will make the required cut at the site that has been recognized in this carrier. All right, then the next will be ligation. So ligation will be done by an enzyme that goes by the name of DNA ligase. So the DNA ligase has a simple role that it will just make the ends to stick together, so that now the desired DNA has now become a part of the carrier DNA structure. So that becomes the next step for us. Then, as I told you, the next process is polymerase chain reaction. And with the help of polymerase chain reaction, we create multiple copies of this recombinant DNA. All right. So amplify a single copy of DNA into thousands to millions of copies once proper gene of interest has been cut using restriction enzyme. And then lastly, we insert the recombinant DNA into the host. There is a name given to this process as well, and this is called as transformation. So the last step here is transformation, and with the help of transformation, ultimately we have inserted the required gene into the plant setup. So this is the entire process. This is the entire process that we use in recombinant DNA technology. Now, so with the help of recombinant DNA technology, we are producing various different types of uh, transgenic crops, and hence these transgenic crops can be useful in various different ways. Now, another application of biotechnology could be biofuels as well. Now, because if you remember yesterday, we discussed about biofuels, and in that we discussed that biofuels basically are what? They made from crop plants, correct? Largely food crops. So because plants are used or crops are used, we can say that this is also an application of biotechnology. So green, it can be considered under green biotechnology. Then after this, there is a concept of biofortification. Now, when it comes to biofortification, this is where we try to fortify the food, meaning that we are trying to increase the nutritional value of the food. We are trying to increase the nutritional value of the food. So increasing the nutritional value of the food simply means that we are adding some component. Usually some component would be either a vitamin or a mineral, because these are some of the nutritions which are required in very trace amounts, but there are regional variations that we see, that there will be some areas which will be good in some kind of mineral or some kind of vitamin, and there will be another area which is better in some other kind of vitamin or mineral. So in this scenario, what we see is that there are regional variations as well. But whatever be the case, wherever we find any kind of deficiency of any kind of micronutrient, then in that case biofortification usually becomes a very important way to solve that problem. So that's why development of micronutrient dense staple crop variety using best traditional breeding practices and modern biotechnology. Now, when we say micronutrient dense, what is the meaning of micronutrient dense? So when we say micronutrient dense, we mean that we are trying to increase the content of micronutrients. Now, there are two ways in which we look at the nutrients, one called as micronutrients and the other one are called as macronutrients. Micronutrients and macronutrients. Micronutrients, the ones that are required in small quantities. All right, these are the ones which are required in small quantity, whereas macronutrients are required in large quantity. Cool. So micronutrients are required in small quantity, and macronutrients, that's why macro, because they are macromolecules and they are required in larger quantities. So in this scenario, for example, vitamins and minerals, vitamins and minerals, they are called as micronutrients, whereas most importantly, carbohydrates, fats, and even proteins, they can be counted as macronutrients. So that's why in this scenario, the ones that are required in larger quantities by the body, they are called as macronutrients, and the ones that are required in smaller quantities are called as micronutrients. If you go to a dietitian, the dietitian will tell you that you have to control your macros if you want to lose your weight. So those macros that they say, macros are nothing but the macronutrients, and they would also say that try to restrict your carbs. So when they say carbs, they basically mean carbohydrates. So these are the terms that we use in today's world. But overall, this is basically what the setup is. So usually what we see is that fortification is not really required for macronutrients, but fortification sometimes becomes very essential in terms of micronutrients. So that is what we do with biofortification when biologically the fortification is being done. So now in this particular scenario, we are basically adding some quantity of nutrient. Even for example, when we talk about fortification, iodized salt that we probably use in our daily lives, that those iodized salts also can be called as uh, fortification. All right. So all these can be counted as fortification. Now, apart from fortification, there is another technique that sometimes is used called as supplementation. So supplementation is when instead of getting the nutrition through food, you are taking some kind of medicine. So for example, let's say vitamin D tablets, vitamin B12 tablets. So all these things are, they can be called as supplementation. They'll be counted as supplementation. Now, in all this, there is one issue that WHO has been talking about, and this is the issue of hidden hunger. Now, what we basically see here in case of hidden hunger is that many a time, especially for the countries and their populations where they are dependent on outside supply of food, they are dependent on uh, imports of food. In these kind of cases, we might also many a time see that although the food supply is made to these countries, the food supply is made to the population of these countries, but many a times they do not get access to a balanced diet, right? A balance of nutrition. That largely we see that something like rice, etc., would be given to them, but which are basically very good sources of carbohydrates. But ultimately what happens is that it's not always that you only need carbohydrates and you need nothing else. So that's why only having carbohydrates is also sometimes not enough, because although it provides the energy that is required, but it will not really help when it comes to the other requirements of the body. And that's why many a times what happens is that although the food supply is being made to these countries in enough quantities, they are making the supply of food, but ultimately what happens is that because of some issues, because of the fact that they haven't been given the exact food that is required, they many a times lose out on the entire balanced diet. So when we say hidden hunger, hidden hunger is seen in terms of supply of food or export of food. So when food is provided to the poor, which may not have every component of the required nutrition. All right, every component of the required nutrition, so that will be called as hidden hunger. That meaning that you are eating but you're not eating enough, you're not getting everything. Now if it's your food choice, then it can't be counted as hidden hunger. It will be counted in terms of wherever food supplies are made. All right, so that also has to be considered here. So there are multiple different foods that we have seen where fortification has been used as a technique, whether it is cassava or maize or sweet potato. In rice and wheat, we definitely do it. In beans, in millets, in all these cases, we have seen that there are various different aspects of fortification that often has been used in all these cases. Then after that, we see that sometimes genetically modified crops are also used as animal feed. That to the animals also the required nutrition many a times is provided through the genetically modified crops. So that these crops will also be fortified with the requirements uh, of the animal. So that even if, let's say, you want to save some cost there, you can ensure that with a limited quantity of food being given to these animals, they can get all the nutrition required. All right, so that is also something that we see. And then there is pest resistant and herbicide resistant cultivars or crops as well, where we know that there are so many crops where pest resistance will be given as a property through some certain genes. Then there are herbicide tolerant crops. So herbicide tolerant crops are the crops where they would have an ability to resist herbicides. What are herbicides? Herbicides are the chemicals, or let's say the pesticides, uh, the chemicals, let's say, which will be used to kill the herbs or kill the weeds that is growing in a given agricultural field. So any unwanted plant, any unwanted growth that has happened in the given agricultural field will be called as herb, will be called as a weed, and to kill that we will be using herbicides. So that's why there are crop varieties that we have been developing which are called as HT variety crops, and these HT variety crops are basically herbicide tolerant. That even if you are using herbicides in the given field, the unwanted plants will die, but the crop which is herbicide tolerant will not be affected. Now we have seen, for example, HT crops also has been in the news mainly because of BT cotton. That there is a variant called as HTBT cotton. Now, the context for HTBT cotton is that HTBT cotton right now is under review with the government of India, and the government of India is looking after HTBT cotton and whether it should be released into the Indian markets or not. Most probably, it will get an approval, and it is expected that this year approval will be given to HTBT cotton. But HTBT cotton was also in the news, or has also been in the news for various different reasons as well. The reason, unfortunately, has been illegal cultivation of HTBT cotton. Especially in two states of Gujarat and Maharashtra, it has been found that there are areas where illegally HTBT cotton seeds are already being sold to the farmers, and farmers have been using it even without knowing in many cases. So HTBT cotton, although right now there is no approval from the government, the approval is pending and most probably is supposed to happen this year, but already HTBT cotton in some parts of the world is being grown. So that is the concern with regards to this particular topic. Then the use of biofertilizers. Now, when it comes to biofertilizers, we know that biofertilizers, of course, are organic compounds. These are organic in nature, and they can have a very good impact on the soil health. So that also will be counted as agricultural application of biotechnology, because there are various organic or biological method that will help us in making them. And then improvement in floriculture. So floriculture, when we say floriculture, means flower cultivation. So from the flowers also there are certain expectations that one has, that you would want the flowers to look beautiful, you'd want the flowers to grow maybe into a bigger size, and maybe all that given, you would want maybe a good scent from from the flower, and all that. You also don't want that the flower just dies down in 1 or 2 days. You would want that the flower continues for, let's say, like 15-20 days or maybe a month if possible. So all these are the qualities that you're looking for in a given flower. Given that scenario, genetic modification can be a big tool for the floriculture industry. Sorry. And that is the reason why we are actually looking for genetic modification in floriculture as well. So overall, what we have seen is that it is not only the food crop that will benefit from this kind of genetic engineering, but at the same time it can also be the cash crops. For example, another case in last couple of years has been of GM rubber. So this GM rubber also is something that will be used or grown in the northeastern states, especially in Assam, but not only in Assam, in other states like Arunachal Pradesh and Meghalaya as well. So in northeastern states, GM rubber is something that we are trying to grow. Now, this GM rubber has been prepared in a manner that it can withstand the climatic conditions and the weather changes that happen in these kind of areas. Because otherwise, rubber that grows in the southern part of the country, let's say in Karnataka and Kerala, then in these kind of cases, we know that the temperature variations are not that great in these areas. They more or less have moderate kind of temperature all throughout the year. So that's why for rubber, those kind of conditions are very suitable. In case of northeastern states, there are certain areas of concern for this kind of rubber, and hence genetic modification in rubber has been done so that we can very easily grow the rubber in northeastern states as well. So overall, we do see that all these are so overall we see that there are all these things that are being done in the agricultural arena. Look at this question. This was asked in your 2011 paper. All right, so I'm launching the poll for you. Please read the question and mark your answer. All right. So as I see, you've marked your answers. So this is a very interesting question from the perspective of the what the answer should be. So many of you have marked uh, C and many of you marked D. Now let me tell you about the question. Other than resistant to pests, what are the prospects for which genetically engineered plants have been created? Question asked in 2011. First, to enable them to withstand drought. This has been done. This we have been doing for a long time. So one definitely is correct. Second, to increase the nutritive value of produce. We discussed about this as well, that biofortification can be done. So this is also correct. Now the problem starts. I mean, let's look at statement number 4, to increase their shelf life. We discussed about this, that this is very often done. So that is also correct. So 1, 2, and 4 definitely correct. Coming to statement number 3, where the problem actually lies, to enable them to grow and do photosynthesis in spaceships and space stations. Whether this is correct or wrong. Now look at the question. Question says, other than resistance to pests, what are the prospects for which genetically engineered plants have been created? Have been created, meaning that if something has been done already, then only you can mark 3 as correct. If it has not been done, then you can't mark it as correct. Now, there is this interesting part. This was not done until 2011. So that's why, if we are looking at this question from the perspective of 2011, Then the answer is 1, 2, and 4. And then the second way to look at this question is, if you are answering this question in 2023, now NASA has been able to do it. International Space Station has been able to do it. So that's why today this is correct. So if the question is asked in 2023, the answer is 1, 2, 3, and 4. So now have been is correct. That's why it's very, very essential to read the questions carefully. When we keep using this very common phrase all the time, cliched phrase, I would say, uh, that read the questions carefully, we really mean it when we say read the questions carefully. And in these kind of questions, as you can see, English matters. That let's say the same question, instead of have been, if the question had said can be, that other than resistance to pests, what are the prospects for which genetically engineered plants can be created? Then can be has no limits. All right, can be has no limits. Then without even looking at the options, just mark 1, 2, and 3. Sorry, 1, 2, 3, and 4, because can be anything basically. So that's why there is no limit to it. So then in that case, we can say that fine. Then 1, 2, 3, 4. Now, but even with have been, when we look at the context in today's world, we know that 1, 2, 3, 4 correct. But since the question was asked in 2011, the correct answer should have been 1, 2, and 4. That's why I said that this question takes an interesting turn in today's world. So any question if you have until now, whatever we have discussed with agricultural application, recombinant DNA technology, any question? What is the concern about HTBT cotton? The concern is that until now, the final approval has still not been given by the Genetic Engineering Appraisal Committee and the Government of India. Although it is more or less expected that the government would give the approval, but the official approval has not been given yet. But there are certain states where we already see that this HTBT cotton is being grown. Some of the reports say that almost 15% of all the cotton being grown in Gujarat and Maharashtra, 15% of that cotton already is HTBT. So although the official go ahead not has not been given yet by the government, illegally already HTBT cotton is being grown in some of the areas. So that is basically what the controversy is about when it comes to HTBT cotton. What are the two states of the two? I mean, there are other states as well, but the number of cases have been found to be higher in Gujarat and Maharashtra. What are restriction enzymes? So restriction enzymes basically are the enzymes that will find out where to make the cut. They will find out matching sequences in the vector genome or the carrier genome, and they will make the cut at the required location so that you can insert the required gene in that location. So that is the role of restriction enzyme, to make the cut at the required location by identifying that required location in the vector genome. Recombinant DNA technology is a type of crafting. Yes, of course. Yes, I would agree with you. It is. CRISPR-Cas9. So CRISPR-Cas9 is a biotechnology, genetic engineering tool, you can say. And this genetic engineering tool has been found to be one of the easiest to implement genetic engineering tool. And what we basically do, there are two parts to it. One is called as CRISPR. One is called as CRISPR, and the second one is called as Cas9. So this CRISPR is very close to how restriction enzymes work. That they would identify and mark the location for gene editing. And in case of Cas9, we say that these are like molecular scissors. These are like molecular scissors. So that's why CRISPR Cas9. All right. So CRISPR basically gene editing tool, and Cas9 molecular scissor. So these two will be used together so that it together becomes a genetic engineering tool, a gene editing tool that will help in cutting out and replacing the DNA, etc., wherever we want. And it has been found to be one of the easiest ones to work with. So there are different types of techniques that we have in gene editing called as SDN1, SDN2, and SDN3, in which we see that in cases of SDN2 and SDN3, these kind of tools can be very, very useful. So that's why we see that CRISPR-Cas9 can become a very important tool to make this entire process much easier. Before this, there were other tools also that we were using. For example, there is another tool using ZFNs. So these tools, although still in use, but they are little complicated to work with. So in those kind of cases, we see that CRISPR-Cas9 completely changed the entire landscape of gene editing by making the entire process much simpler compared to what until now it used to be. So, please add CRISPR-Cas9 to ARM. I think it will be there, although I don't really believe why CRISPR-Cas9 I don't teach anymore is mainly because it has been asked a couple of times in the examination already, and now it's not very important as a topic anymore. So although it has become very famous as a genetic engineering tool, but I would still say that it's not a very important topic. It was a very important topic for a couple of years between 2015 to 20, I would say. It was a very, very important topic. Not really anymore. So that's why the portions where I used to teach CRISPR-Cas9, I have taken away from the class. All right. So next we have, so any other question, anyone? ZFN. ZFN is nothing but another genetic engineering tool which is used for cutting and replacing the DNA, just like CRISPR-Cas9. It is but little complex to implement, so that's why CRISPR-Cas9 technique has been found to be better than the ZFN. All right. So moving on. I hope no other question. Hmm. So let's move on. So next topic for us is white biotechnology. Now, in case of white biotechnology, we are talking about industrial application. So whenever industries use biotechnology, it is called as white biotechnology. Now, again, there are various ways in which industries could also use biotechnology. For example, the process of fermentation, which is used to make alcohols. Alcohols become very, very important reagent for a lot of industrial processes, and in many different industries, alcohols as an organic molecule would be used in one way or the other. So that's why the process of fermentation becomes very, very important from that perspective. The same fermentation process could also be used for the production of bioethanol. Yes, we discussed about this yesterday, that bioethanol basically is produced out of the process of fermentation. So that's why this entire process of fermentation, organic fermentation, can be very, very crucial, and that's why in industries it finds a very, very important role to play. Then apart, so one is about fermentation. Then another way is to enhance the oil recovery from the well. That many a times we see that there are ways in which genetically modified organisms, they would be able to consume. So if you see here, consume dense hydrocarbons and reduce the surface tension of the oil to a great extent, and hence facilitate easy recovery of oil. And especially in the areas which are dilute, especially when it comes to dilute environments. So when I'm saying dilute environments, it means that wherever the presence of oil is less. Because if you have abundant presence of oil, then the chemical processes, the industrial processes of oil extraction can be used. But when it is not that dense, when the concentration is very low, it's a very dilute condition altogether, then in all these kind of cases, that what we'll see is that genetically modified organisms can be used, and they would be able to eat upon the heavy hydrocarbons, and then you can extract these hydrocarbons or extract this oil from these microorganisms. So in this manner, it becomes a cost effective way. Otherwise, if we are using the industrial way for a very dilute environment, it will not be cost effective. So that's why we avoid using those techniques in dilute environments, and instead we could use biotechnological methods here. So one way is that, and then after that they can also act as preservatives as well. So that is also one of the ways in which we can make the use of biotechnology in the industries. Then apart from that, there are certain industrial processes as well, where we see that this process of biotechnology will be used by the industries. One such process is biosorption. Now, biosorption, if you try to break down the word, you'll again find that there are two words that you have in a manner. You can say that this is made of biological absorption. Biological absorption, that's a biosorption. So what we do in biological absorption is that this is where whatever dilute especially, let's say, let me put it this way, that whenever we have any industrial unit, any factory, etc., the contamination of the land in the nearby areas could be high because of whatever chemical procedures are being used in these kind of areas. So now what happens is that because these areas become contaminated, polluted, etc., that is where microorganisms will be used in a very similar way how we described the recovery from oil wells. But this time it is not about discovery. This is not about recovery of oil. Instead, it is about getting rid of the contamination. The contamination that might have been that might have built in a given area, that we want to remove. All right, that we want to remove, and that is why it is called as biosorption. So overall, this is basically what we'll see with biosorption, that the process is very similar to what we do in oil recovery, but this time that was being done to get the oil. This time it is being done to get rid of the uh, uh, waste, or get rid of the contamination. So all these heavy metals, etc., they could be bound by these microorganisms and then taken then away. Then we have biomining. So biomining is where you are using any kind of biological resource for mining purposes. So for example, what we discussed in oil recovery, similarly mining of various metals, etc., can also be done. Not only that, and it's not only genetically modified organisms or microorganisms, etc. At the same time, even there is something called as phytomining. So phyto, when you see this term phyto, wherever you see this term phyto, understand that. Can you tell me what is the meaning of the term phyto? Where do we use the word phyto? To represent what? Yes, plants. Perfect. So phyto is used for representing plants. So when we say phyto mining, it basically means that we are talking about mining with the help of plants. How exactly? That this time what we see is that the roots of the plants, they can actually absorb some of the heavy metals from the soil, and then from there on, from the roots of these plants, we can just burn up the root, and from the root, the ashes of that combustion, we can actually extract the metals, or extract whatever we are there for. So that is called as phytomining. So if the mining is being done naturally with the help of plants, it becomes phytomining. So it becomes a form of biomining in that sense. Then after this, we have blue biotechnology. So blue biotechnology is nothing but when we are talking about aquatic environments, that when biotechnology can be applied to aquatic environments, it will be called as blue biotechnology. Now again, there are various applications, like for example, whatever you're doing on this, in the water or maybe on the surface of water, will basically become blue biotechnology. So in this, there is one important application, and this is from the perspective of the oil spills that happen many a times. So in case of oil spills, what we see is that there is a particular formulation of bacteria called as oil zapper. Oil zapper is a mixture of 5 different types of bacteria, and these 5 different types of bacteria, they feed upon the hydrocarbon presence because of the oil spill, and ultimately it just breaks down the hydrocarbons into carbon dioxide and moisture. So in this manner, they are able to eat upon all the organic buildup that might have happened, or the oil that would have spread to the sea or the ocean surfaces. So that is where oil zapper actually works, which is nothing but a mixture of 5 types of bacteria, which can actually convert hydrocarbons into carbon dioxide and water. So in this manner, we understand that there are various aspects of the use that we have for biotechnology in underwater applications as well. Then the next one is how biotechnology helps the environment. Now, in this particular scenario, there is a concept called as bioremediation. So bioremediation is whenever we are trying to clean up the environment with the help of some biological resource, meaning that if we are, for example, let's say uh, we are cleaning up the environment with the help of a microorganism, with the help of a plant, then it will be called as bioremediation. Now, bioremediation can be of two types. It can be an in situ bioremediation or an ex situ bioremediation. In situ, you might have heard of this term in environment also when we say in situ conservation and ex situ conservation. So in situ and ex situ. In situ means at the site. In situ means at the site and ex situ means away from the site. In situ means at the site and ex situ means away from the site. So when the bioremediation technique, that is the cleaning of the environment, is happening at the site of contamination itself, then in that case it is in situ bioremediation. But if we are taking the waste or we are taking the contamination away from its original location, treating it somewhere else, and then maybe bringing them back, then in that case it will be called as ex situ bioremediation. So so that's why we have to understand that whether wherever the contamination is happening, if we are doing the remediation at that point itself, then in that case it will be called as in situ, otherwise ex situ. Now first, to start with in situ, we are saying that in situ is the treatment of the waste at the site of the contamination itself. Now, overall, there are various techniques that are used. For example, there is something called as biosparging, bioventing, bioaugmentation, and bioculture. These are some of the in situ bioremediation techniques that are very often used. So let's try to understand what do we mean by this. So first one is biosparging. Now, when it comes to biosparging and bioventing, these are two concepts, or these are two methods that are very similar to one another with one small difference. So understand what is biosparging and what is bioventing. So suppose we are talking about a layer of soil. We are talking about layers of soil, and in this layer of below the layer of soil, water starts. This is where it is near the water table. Now, in this scenario, if we consider these two parts of the soil, if we consider these two parts of the soil, now the part that is near the water body, of course, will have higher moisture content. Understood? It will have higher moisture content because it is near water body, and the other one, it is away from the water body, so it will have lower moisture content. So this part is called as unsaturated zone. This part is called as unsaturated zone, and this part is called as saturated zone. So unsaturated zone and saturated zone. So unsaturated zone because it has less moisture. Unsaturated zone because it has less moisture. And saturated zone because it has high moisture content, as it is near the body of water. All right. So this is what is called as unsaturated zone and saturated zone. Biosparging is done in this saturated zone. All right. Biosparging is done in the saturated zone, and bioventing is done in the unsaturated zone. Correct. So biosparging is done in the saturated zone. Bioventing is done in the unsaturated zone. So in this particular scenario, when we are looking at these two aspects of saturated and unsaturated, what we do in these two cases, just understand this, that microorganisms are already present in these areas. Soil will have its microorganisms. So microorganisms are already present, but they are working in their natural pace, which is slow. All right, they are working in their natural pace, which is slow. We want them to work faster. We want them to have an effect faster. So for doing that, for making them work faster, we provide some extra nutrition. We provide some extra nutrition, oxygen, etc., so that they get that energy, they get that nutrition, and they start working quicker. So this is what we do in both the cases of bioventing and biosparging. All right. So, but the difference that we will have is that when we are going to the saturated zone versus when we are going to the, sorry, when we are going to unsaturated versus saturated, going to saturated zone will be easier. Why? Because it's just like at the surface. So that's why going to the saturated zone, unsaturated zone, injecting the nutrients and oxygen in the unsaturated zone will be easier, whereas injecting the nutrients and oxygen uh, at the saturated zone will be difficult, because you have to go deep inside, so you need high pressure. So that's why this will be done at high pressure. This will be done at high pressure, and this can be done at low pressure. All right. So bio so so one can be done at high pressure, one can be done at low pressure. So that's why this is where we do biosparging, and this is where we do bio venting. So this is how so what we see is that air or oxygen and nutrients are injected at high pressure to increase the biological activity of the indigenous microorganisms and to enhance the decomposition activity. So that is what I was saying, that naturally also decomposition process would have taken place, but they would have taken more time for that decomposition. But when you are adding additional oxygen or nutrients, etc., the energy that the microorganisms would receive, that will help them in fastening the process of decomposition. So that is basically what is happening in biosparging. The same happens in bioventing as well, just that this time it is something that will be done in low pressure, at low pressure. All right. So same thing, oxygen, nutrients, etc., will be added to this layer, but this can be done at low pressure because it is just at the surface itself, so it's easier to carry out. So that is called as bioventing. All right, this is what bioventing is. Then, looking at the ex-situ bioremediation techniques, there are two important ones, one called as land farming and then another one called as biopile. So in case of land farming, it is nothing but a periodic tillage of soil. Periodic tillage of soil so as to increase the activity of microorganisms. So in case of this ex situ bioremediation technique with the help of land farming techniques, what we basically see is that land farming, I mean the contaminated soil will be taken away from the farm. Then after that, periodic tillage of the soil will be done to ensure that we are able to enhance the activities of microorganisms in the soil. So it's basically saying that the microorganisms have slept and we are now trying to wake them up with the help of tilling the soil. So once they wake up, they'll start the decomposition process faster. So that's why the tillage process is used in land farming. And then after that, we have biopile. Now, in case of biopile, we say that it is a hybrid of land farming and land composting. Because in such a scenario, when it comes to biopile, in biopile we try to take care of each and every feature of that soil. What should be the moisture content, the temperature, what kind of alkaline or acidic soil is required, how much of nutrition, what is the pH value. All these things are controlled so that we can enhance the biodegradation process, and all these will add to the value of the soil to ensure that what exactly was the nature of the soil that somebody required. Accordingly the soil can also be made. So in case of biopile we see that composting techniques and land farming techniques both are used at the same time. Typically the treatment can take up to 3 to 6 months of time. It obviously is a time taking process, and it will be kept very checked very closely, and we keep on working on it, and ultimately it is to the required sent to the required agricultural field. Then there are some other bioremediation techniques as well. For example, we have phytoremediation. So phyto, we discussed that it is plant. So when we say phytoremediation, it means that we are trying to solve the problems with the help of plants, that we are trying to clean the environment with the help of plants. So that will be called as phytoremediation. Similarly, there is something called as phytoextraction. So phytoextraction is when the plant is capable of removing all the harmful chemicals or the harmful metals, etc., the harmful compounds also, or heavy metals, whatever it is. All these things, they can actually be removed from the soil with the help of the plant, with the help of a plant, and hence we can make the use of phytoextraction technique as a bioremediation technique. So in this manner we see that phytoextraction can also be very useful. So phytoremediation and phytoextraction, you can think of it as uh, um, as a sub process of phytoremediation. All right. So and plants, normally we, you can simply say that all the plants are doing phytoremediation for us, right? So for example, we know that the plants will take in carbon dioxide and release oxygen. So in a manner they help in purifying the required location, or in any given location wherever we are. So that's why that naturally, anyway, we know that plants help in phytoremediation. In general, plants behave in a manner that phytoremediation is possible. But there are special techniques, and there are some plants and trees. For example, neem tree is known for purifying the given surroundings much better than many of the other trees. So that's why it counts in phytoremediation. Then after that, lastly, we have mycoremediation. By mistake, it has moved there. It is mycoremediation. So when you see is something related to the fungus, or what we, in plural form, we call it as fungi or fungi or fungi, whatever you want to call, doesn't matter. That's why I'm using the same safe word, that is fungus. Otherwise the it can be called as fungi or fungi or fungi in plural form, correct? So so when we are using this fungus in a remediation process, it is called as mycoremediation. So in all these cases we see that these are the various techniques of bioremediation which are used in cleaning up the environment. Look at this question. This question was asked in your paper. All right, so I hope uh, you have read the question. I'm launching the poll for you. Please mark your answers quickly. Hmm. So 6% A, 24% B, 41% C, and 29% D. All right. In the context of solving pollution problems, what is or are the advantage or advantages of bioremediation techniques? First point is, it is a technique for cleaning up pollution by enhancing the same biodegradation process that occurs in nature. This is correct. This has to be correct, because what did we just go back and think of what did we do? For example, in case of biosparging, we said that microorganisms would have done their job, would have done the breakdown or decomposition that is required, but we want to enhance that process. We want to enhance that process, and that is what we are trying to do. So that's why this is definitely correct. One is correct. Second is, any contaminant with heavy metals such as cadmium and lead can be readily and completely treated by bioremediation using microorganisms. Now look at this particular question. Sorry, statement. Any contaminant readily, completely. These are all what? Extreme words. Three extreme words in one single sentence, in one single statement. This can't be correct, right? So anyway, when it comes to heavy metals often are not uh, cleaned up by microorganisms, but rather we use plants in these cases. The phyto extraction techniques. Phyto extraction techniques are more used in these kind of cases, right? Because when it comes to the structure of all these heavy metals, it's not easy for microorganisms to act on it or to decompose them. So that's why in these cases we see that phyto extraction techniques or phyto remediation techniques work much better. So that's why when it comes to heavy metals, we will use phyto extraction will use phyto extraction and will not use microorganism. And that's why any, readily, completely, all these are very strong words. And then, so one is correct, two is wrong. Genetic engineering can be used to create microorganisms specially designed for bioremediation. We can do that. We can create microorganisms which are genetically modified in a manner that they will be more useful for us. So for example, if you think of oil zapper. So in case of oil zapper, we can say that it is where we have created microorganisms especially for bioremediation. So that's why 3 is correct. So correct answer will be C. That is 1 and 3 only. These are the only two statements which are correct. Another question was asked. This is in the context of oil zapper. I'm sure you know the answer. It's a very simple question. Please mark your answers quickly. All right. So, as I see, 53% of you have marked A. Only 53% of you have marked A. What is oil zapper used for? We discussed that wherever you have oil spills, we can use oil zapper. Look at the first option. It is an eco-friendly technology for remediation of oily sludge and oil spills. Things can't be clearer than this. It is the latest technology to develop for undersea oil exploration. It has nothing to do with exploration. It is about waste management. It is about clearing the contamination. So then, in that case, B is not possible. I'm surprised that so many of you have marked B, and even C has been marked by some of you. It is a genetically engineered high biofuel ending yielding maize variety. Where did we discuss this in the context of oil zapper? Latest technology to control accidentally caused flames from oil well. It's not about the flames or the fires, it's about oily sludge and oil spill. Frankly, I was expecting much better response than this. This was an easy question because we just discussed that oil zapper can be used in treatment. It can be used wherever oil spills have happened. Oil zapper will go. They will be. It's a formulation of 5 different bacteria set to that location, starts eating up on the hydrocarbons and just clears off the hydrocarbon. So you'll have to concentrate better than this, yeah. You'll have to do better. Uh, I needed more correct answers in this question. So, any question, anyone, with regards to whatever we have discussed until now? Can heavy metals be completely and readily treated by phytoremediation? No. When we say completely and readily, these are very strong words, and heavy metals in general, removal of heavy metals is not that easy. So yes, phytoremediation techniques or phytoextraction techniques to be exact are used in these cases, but still saying readily and completely would be still very strong. So that's why that's not possible. How can I remember all the things? It's simple. Revision. Revise well, look at the words, understand the meaning of the words, and you will be able to do that. It just needs a couple of revision from you. It's not a very difficult topic that we are reading, just that it needs some practice. All right. So next topic for us is cloning. So until now we have looked at various branches and how these various branches are using biotechnology in one way or the other, and we see that there are immense number of ways in which we are using the process of or any process related to biotechnology. Now after this, the next one, in the next segment that we are discussing here, we will look at some of the specific topics. All right, there are some of the specific topics which are very widely discussed in the context of biotechnology. So these are the topics. So first of those topics is cloning. Now, when we look at cloning, understand what the meaning of the term cloning is. So, cloning is a process where we say that it is a process of asexual reproduction in which the offspring or the progeny is an exact replica of the single parent who has contributed to the genetic material. Meaning that cloning as a process, what we see is that cloning as a process is called as asexual reproduction. Now, why are we saying that it's an asexual reproduction? It's an asexual reproduction process. It's mainly because of the fact that when it comes to asexual versus sexual reproduction, in case of asexual reproduction, there's always only one parent which is involved, and there is no interaction between two parents. When it comes to sexual reproduction, it is where we have interaction between two two organisms. One may be male, one may be female. So it is an interaction between the two, and this interaction between these two will ultimately lead to the reproduction of or the birth of a new progeny or offspring. So asexual versus sexual. So this is basically what happens, that in asexual reproduction, the genes will be entirely from one individual. Gene from only one individual. And by individual, it could be an individual animal, individual plant, whatever you want. And in case of sexual reproduction, we see that there will be a genetic mixing among two individuals. So because of that, what happens is that the offspring born out of sexual reproduction will be unique, because the genes from both mother and father have combined, and there is a unique set of genes that have been produced. So it will definitely share many traits with the father and will share many traits with the mother, but ultimately it will be unique in the sense that it is a combination of two genes. Whereas when it comes to asexual reproduction, there is only one single parent, and this one single parent has contributed to all the genes that this organism has. And there are so many techniques. So for example, let's say if you consider cell division that happens in many of the small microorganisms, simply what happens is that the nucleus starts to divide into two parts, and this nucleus, once it has almost started to divide, it basically divides cells into two parts. So that is called as cell division. So everything that these two cells have basically comes from the same cell that existed, and that's why it is called as that's why there is a complete transfer of gene. Similarly, budding as a process, in case of budding also, you'll see that a small bud will be formed on the surface of the organism, and the bud starts to grow, and then ultimately it detaches, right? It ultimately detaches and grows into a new individual. So all these are the processes which will be called as asexual reproduction. So normally we see that the lower plants and animals, uh, even microorganisms, they normally would show asexual reproduction, and the ones which have very complex genetic structure, higher plants, higher animals, humans, etc., they would show sexual reproduction. Now in case of cloning, we are saying that we are producing an exact replica of one parent that has contributed to the genetic material. Now, this becomes a unique process because we are saying that we are going against the natural order of what happens in the higher plants and animals. But and that's why the term cloning, when we use, very often cloning is used for creating a copy of something. You'll say that you have cloned something. So now in this particular scenario, what we basically see is that cloning, that's why it will be called as a process of asexual reproduction. Now, in case of cloning, we have seen that there are cases of animal cloning that we have used, and this animal cloning can be very, very useful in various different ways. Now, for example, let's say when we see an animal that is stronger, that has better genes, we might want to replicate those genes, and that's why we see that cloning will be carried out from this kind of an animal. So wherever we are looking for some healthy traits, some positive traits, we can make the use of cloning as a process. So overall, that's why cloning can be used in animals. Now, animal cloning has been in news and has been in use for last like 25 years. This picture that you see right here, this picture was the first successful case of animal cloning, first successful case of animal cloning. And the name of this sheep was Dolly. So Dolly the sheep became the first successful case of animal cloning. But getting this success in animal cloning also took us a while, because animal genes are much more complex as compared to plant genes. So that's why, although in plant genes we have had certain successes, we have seen fair amount of success, rather, I would say, in all these cases, but that hasn't been the case when it comes to animals. And even in this first successful case that we saw, it took us 277 attempts. This was the 277th attempt in getting that success. So just understand the value, that how difficult this process has been, that it took us 277 attempts to be successful finally. How many attempts do you get in this examination? So I know, a painful question. So that's why in this particular scenario, we see that this is how cloning basically works. Now, when it comes to the process of cloning, before we go on there are some examples of cloning that has happened in India as well. So, for example, Samrupa was the first case of animal cloning in India, and in 2009 it became world's first cloned buffalo, although did not live for very long. It just lived for almost like 5 years, 5 days, and then it lost its life. It succumbed to lung infection, just 5 days after it was born. Then after that, the next was Garima, second cloned buffalo, lived for more than some 2 years, but then again because of heart failure, died in 2011. So this is something that we have seen, that in many of these cases, uh, the clones have not lived for very long. The clones have not lived for very long, and we have seen that they have succumbed to some kind of infection. They have died, and that's why there are conditions that are maintained for animal clones many a times, that they'll kept isolated, they'll be kept away. A lot of antibiotics, antivirals, etc., will be given to them so that they can survive. So that is the problem that many a times we see in case of animal cloning. Now, apart from that, there was another attempt, and this was CIRB Gaurav in 2016. This was done. Then, apart from that, there have been other attempts as well. For example, until now, if I'm not wrong, there are total of 26 cloned animals that India has altogether. This was a recent example, not very recent, almost 2 3 years back, where we very where we for the first where we have cloned all these animals. So if you see, this was a recloning of Hisar Gaurav or CIRB Gaurav, and there are 7 clones of M29 Murrah bull that we have produced through the process of cloning. Apart from that, we have seen that this year also, 2023, March 2023. March 2023, there was the latest incident of animal cloning, and this basically was a clone of Gir cow. So we saw that a first clone of a cow in India has been done. So this becomes first cloned cow in India. So that's why we have seen that all these are some of the aspects that we are looking at. Now, overall, we see that why, if you ask me why, for example, let's say when it comes to Gir cow, that why did we do a cloning of Gir cow? So generally what we see, that these indigenous animals, they have better resilience towards the conditions that prevail in India. So for example, we see that they are better resistant, so they are, you can say, more disease resistant, they are also adapted to the hot and humid climate. So that's why we see that there are all these things that we have. So overall, they will have better potential, and we'll see that they will help in, it was said that it will probably help in better milk production. So it can be used in these kind of cases, can be used in enhancing milk production in the future. So overall, we see that there are all these cases that we see uh, of cloning that has been done in India. Now, when it comes to the process of cloning, we see that the process of cloning starts with something called as a somatic cell. So somatic cell means any cell in the body that is non-reproductive in nature. So for example, in humans, there are two cells that can be called as reproductive cells: the sperm cells in males and the egg cells in the females. These two cells are called as reproductive cells because these are the two cells that participate in reproduction. Apart from these two, all the others are called as somatic cells. So first and foremost, for the cloning process, we extract a somatic cell. All right, we extract a somatic cell. Look at this particular picture. We first extract the somatic cell, and from the somatic cell we isolate the somatic nucleus. we isolate the somatic nucleus. So this somatic nucleus basically will be isolated, will be taken away, and this will be inserted inside an enucleated egg cell, meaning that we take an egg cell, from the egg cell we take away the nucleus, and this is replaced with the somatic nucleus. So now you have the egg cell with somatic nucleus. Now if I ask you why, can you take a guess that why are we doing this process where we are removing this nucleus and we are adding this nucleus? Why are we doing that? Just think of an answer. Any suggestions are welcome. What do you think? Why are we doing this? For parents' DNA, no, not exactly. Nucleus carries genetic information. Perfectly correct. This actually is the answer, that we discussed about this, that all the genetic information, all the specific characteristics that a person has, all these things are there inside the nucleus, right? So that's why we need the nucleus. We discussed that nucleus is the brain of the cell, correct? It is like the brain of the cell. So it has all the information about that individual, all the activities, etc., about the individual. So once you have taken away the nucleus, you basically have taken away the brain of that individual. So nucleus will be taken away, and this nucleus will be inserted in the egg cell. Because if you had the nucleus of the original egg cell, it would simply mean that then this egg cell will have the individual characteristics of this egg cell. But you don't want that. You want the clone to have the exact properties of this particular person, or even if it was an animal. So then that's why you will take the nucleus, insert inside the egg cell, and then activation will be done. So with activation, we see that embryonic development starts. This is something which is called as blastocyst. This is called as blastocyst. So now in case of blastocyst, we see that blastocyst is nothing but a bunch of almost 150 undifferentiated cells. And this activation has been done with the help of some electric shocks and some chemicals. So with the help of that, we see that activation is done, and very quickly, within a week's time, let's say, there will be a bunch of almost 150 undifferentiated cells. So this is called as blastocyst. Now from here on, there are two things that we can do. Either we can insert this inside the uterus of the host mother, or it can be taken away in the lab. So if it is for the reproduction, if it is for the birth of a new individual, then it will be called as reproductive cloning. If that's not the case, if it is for some kind of medical procedure, then in that case it will be called as therapeutic cloning. So that's why there are two types of clonings that we have. One called as reproductive cloning, second one is called as therapeutic cloning. So depending on what the requirement is, we see that one it can be either this or that. So cloning is performed by somatic cells, nuclear transfer into the enucleated cell. The eggs so obtained thereafter simulated by electric shock and chemicals to initiate division. Within a week, a single cell becomes a ball of mass having around 150 unspecialized cells. And then ultimately, this blastocyst could be inserted inside the uterus of the surrogate mother to complete the process of reproduction. Otherwise, if we want, the same can be also sent to the laboratories or to the research center, so that they can do any kind of study or research whatever they want. So that's why reproductive cloning, the newly created embryo is placed back into the uterus, where it takes time to develop into an individual. Reproductive cloning is the production of genetic duplicate of an existing organism, correct? And in case of therapeutic cloning, we basically see that we try to harvest stem cells from here. What do we mean by stem cells? We'll discuss about that in some time. So this is what we try to do in this particular scenario. This is a question that was asked in your paper. Very simple question. Read it carefully. Please mark correct answer for me. Wonderful. So 81% of you have given the perfectly correct answer, which is C. And yes, 14% of you did get stuck with D, production of organisms free of disease. So see, saying that something will be free of disease is very extreme. That you might say that, okay, fine, I am doing the process of cloning because I want to avoid this certain disease or that certain disease. But saying that something will be born free of disease can be a little extreme. So correct answer will be reproductive cloning of animals. So somatic cell nuclear transfer, because we are taking the somatic cell and we are transferring the nucleus, and then that's why this technology is also called as somatic cell nuclear transfer technology. Then lastly, there are certain concerns also that we have over reproductive human cloning. That first and foremost, there are chances that it might undermine the society's respect for human life. It might so happen that tomorrow, if process of cloning is being used widely, then there will be a clear demarcation between perhaps what is clone and what is an actual human, and then we might start a kind of a divide between the clones and the naturally born humans. So that's why maybe they'll start getting treated as secondary race, which is not going to be good for the fabric of the society all across the world. It may affect the social institutions such as marriage and family. You might think that, why do I need to get married? Why do I need a family? I can clone myself and get an offspring. So why to go through the entire process also? Maybe somebody who's a fan of themselves might feel something like this. But overall, we do see that these things can become a possibility, because single parents can very easily go to uh, single parents can very easily think of this instead of trying to get into a family situation. Then it can create global security concern as well, that we might see that some of the countries or maybe terrorist organizations, they start with the process of cloning so that 10-15 years later they have a cloned army, and then they don't have to send their soldiers, instead of that send cloned army. So that also becomes little problematic. Then reproductive cloning may emphasize on infusing desirable traits in clones, and this may promote the concept of designer babies, which are genetically engineered to exhibit desirable characteristics. Now, when it comes to this concept of designer babies, uh, the name tells you a very unique sounding term. But what happens in case of this designer baby is exactly what we are doing, that we are designing our children. We want to design our children, meaning that we know that these are some good genes or these are some bad genes. So we want to have a child which has only good genes and no bad genes, so that they would be much superior than anyone else that we have. So that becomes a designer baby, that becomes the concept of designer baby. So that is why this also can become a reality when cloning is allowed, if cloning is very easily allowed to happen. So that is also one problem that we might face. And then one major problem is also related to the morals, that where we can say that many a times what happens is that these embryos on which we are working, or wherever we are using it for therapeutic purposes, medical purposes, every time research or study is done on an embryo, we simply are killing a life, because we are not going to let it grow into an embryo and from there on grow into an individual. Instead, we perhaps in the course of our study and research we might kill it. So that's why that also is not acceptable according to many cultures, and goes borderline into the immoral side of the story. So that's why there are all these issues that we have with cloning altogether, with especially with human cloning. And that's why you will see that human cloning has not really been allowed by many of the countries. That many countries have simply banned, or rather every country right now has banned the direct use of human cloning. Only under extreme circumstances, therapeutic cloning in certain cases might be allowed. Now then, one more thing in this topic, what you should understand is that there is a basic difference between reproductive cloning and surrogacy. That although in case of surrogacy also there is an outside external mechanism available, but essentially these are two completely different processes. So for example, in case of reproductive cloning, we see that the process is that you are taking the somatic cell and you are replicating it, whereas in case of in vitro fertilization or test tube baby or surrogacy or whatever you want to call it, in this particular case we see that there is still an actual fertilization that happens between two parents, male and the female cells, instead of that happening only in one cell. So that's why the process is different, that one has two parents involved and one has only one parent involved. Then after that, uniqueness of progeny. Now uniqueness of progeny also is a factor, because in case of in vitro fertilization, because it is a normal sexual reproduction, there will be the uniqueness that we see in the progeny or the offspring. But in case of cloning, what we see is that it's an exact replica, because there is only one set of genes that has been transferred from the parent to the child. So that's why there is no difference here, that's why there is no uniqueness that comes in into the child. Then one major, very major problem is with the lifespan. We saw some of the cases of animal cloning, and what we understand there is also the fact that animal cloning or animal clones have not lived for very long. Correct? They have not lived for very long, and very often we have seen that they have died some days later, some months later, or maybe a couple of years later, but they do not lead a long and healthy life. That again is not the case with in vitro fertilization, because there we see that they would lead a long and healthy life. There is nothing different from the natural process of fertilization technically. So that's why we see that that's a completely different story. And then the ethical issues that we discussed, that when it comes to reproductive cloning, there are ethical issues that we need to address. But when it comes to in vitro fertilization, these kind of issues do not make any uh, do not create any problems. That's why there are no issues whatsoever when it comes to ethical issues in vitro fertilization. So essentially these are two completely different processes. That in vitro fertilization is sexual reproduction, whereas cloning is asexual reproduction. So that changes the entire landscape of the offspring as well. So cloning, just remember that cloning as a process, although animal cloning can have some advantages for us, the strength of the animal or maybe other good qualities or good genes that might be present in that animal, but at the same time we need to understand that when it comes to cloning, and if let's say the governments, that what they have done right now is that they more or less have banned the use of cloning. But if it is allowed, we don't know what direction the society can take. Maybe nothing will happen, but maybe there are a lot of negatives that will build from here. So that's why there is a very close watch on any kind of cloning that happens anywhere in this world, especially when it comes to human cloning. So that's why these are some of the areas of problems. Next, we have stem cells, but let's take a small break here. It's 3:42 right now. Let's take a 10-12 minute break. Maximum, please come back by 3:55 p.m. And then after that, let's proceed. And the next topic for us will be stem cells. Welcome back, everyone. So first, if you have some question, if you want to ask, please let me know. In human cloning, exact offspring of same physical and facial characteristics are formed. See, you can't really guarantee that 100% everything will match, because not everything is genetic in nature. Something is also dependent on your growth, your nutrition, and everything. So that's why while the growth of that individual happens, there are certain changes that may happen. So that's why it's not necessary that every feature will remain exactly the same, even though the genes that the individual was born with was exactly the same as that single parent. All right, so let's move on. The next topic for us is stem cells. Now, in case of stem cells, what we basically are saying is that stem cell, just understand the value of stem cell. That stem cell, we say that these are the raw material. These are the raw material for any cell type, meaning that any cell that we have in our body, they are basically formed from stem cells. Just like think of how the birth of a child happens, that before the child is born, it would first be just a fetus, an embryo, and then we see that this embryo starts to grow. And while the embryo grows, this is where the various organs in that embryo also start to grow. And ultimately, by the time it is ready to be born, we know that all the different organs in the body of that uh, individual, or in the body of that uh, uh, embryo, would have grown. So similarly, what we see is that any cell that we have, you know that a basic cell would have nucleus, cytoplasm, cell membrane, and all the different organelles like mitochondria, ribosomes, etc. There will be all these things that we have in a cell. But before it specialized into a cell like this would not have been just a cell, but it would have been an undifferentiated cell. An undifferentiated cell is called as stem cell. So in a manner, we say that stem cell is the raw material. All right, stem cell is the raw material for any given cell. So then in that case, so that's why we are saying that these are the raw material for other body cells, and they are considered raw material because all the cell with specialized functions are generally are from these cells. So that's why stem cells can become anything. That's why this meme that the cell stem cells can become anything. So that's why when if I'm asked to become anything, I'll become a stem cell so that I can become anything. So stem cells will have an ability that they can specialize into all this body cell types, and that's why they become very, very special. So first thing to understand, that these are the raw material, correct? Stem cell is nothing but the raw material for any cell type. All right, now from here on, what we also have to understand is that, in other words, stem cells are unspecialized cells which have not yet developed into mature specialized cells. All right, they have not developed yet into mature specialized cells. So as the step of development happens, the stem cells will convert into some kind of stem cell, or sorry, some kind of body cell. Now overall, when it comes to the stem cells, we see that there are various different types of stem cells, uh, and all these stem cells will grow into some specialized cells. So for example, all these can be called as specialized cells, like you have the nerve cells, the blood cells, all these muscle cells, etc. So all these basically are the different types of cells, and they would all grow from stem cells. So stem cells, while they start growing, they would first grow into, they would keep on dividing, and cell division will keep on happening without getting specialized. So there are multiple copies of stem cells that will be produced first, and then they start to specialize. And once they start to specialize, they would specialize into all these different types of body cells. So overall, what we see is that when it comes to stem cells, overall, they basically have two important properties because of which we are actually even studying the stem cells. So two important properties that we have is, first, their ability to self renew, their ability of self renewal, and then their ability to specialize. So understand what I'm saying, that stem cells, we are saying that stem cells will grow into any kind of cell, correct? But before growing into any kind of cell, they would keep on increasing their number. Cell division, multiple cycles of cell division will happen before they start to specialize. So that's why we have number of copies of stem cells available with us. So that's why their ability of self renewal, that ability of stem cells to go through multiple cycles of cell division while maintaining the undifferentiated state, is called as self renewal, the ability of self renewal. And then after that, the ability to specialize, that we know that they can differentiate into various different specialized cell types. So in this particular manner, we see that this is the ability that they have to specialize into any kind of cell. Now, these two properties make the stem cells very, very special, and that's why they would be able to help us in various different types of applications as well. What are these applications? We'll come to that, but before that, one more thing: the source of a stem cell. Now, sources of stem cells can again be varied depending on when have we derived the stem cells. The first one will be called as embryonic stem cell. When you have extracted the cell in the embryonic stage, it will be called as embryonic stem cell. So in the embryonic stage, we of course know that the embryo has not developed any organ, any part of the body, etc. So then in that case, because embryo itself is in an undifferentiated state, the stem cells also that you get from here, they have a better ability to specialize. So depending on what the origin of the stem cell is, whether it is a stem cell from a fully grown human, let's say like me, or whether it's a stem cell from an embryo where none of the body parts have yet developed. An embryonic stem cell will have a better ability to specialize as compared to another stem cell, which is called as adult stem cell. So stem cells derived from the undifferentiated inner mass cells of a human embryo. This will be called as embryonic stem cell. So an embryo is called as blastocyst and has around 150 cells. Now, apart from this, there are adult stem cells. So adult stem cells, these are basically found in the adult tissues such as bone marrow or fat. Now in this case, when it comes to adult stem cells does not mean that you have to be of a certain age to have the adult stem cells. Adult stem cells will be found in anyone who has born. All right, when the child has born, after that the cells that are found are called as adult stem cells. When all the organs and the organ systems, etc., they are fully grown, then after that the stem cells that we have are called as adult stem cells. Now the problem with adult stem cells is they have a limited ability to specialize. In case of embryonic stem cells have a much better capacity to specialize, because we have embryonic stage, we have them in the embryonic stage. Hence they'll be able to specialize a lot better into a lot more different types of cells. Whereas when it comes to the adult stems will not be able to specialize into all the types of cells. So they will have a limitation when it comes to their ability to specialize. So that is what we see with regards to the adult stem cells. Now, one more source is perinatal stem cells. So when we say that something is a perinatal stem cell, it means that it is being derived from the perinatal tissue sources, such as the umbilical cord or placenta. So in all these cases, amniotic fluid. So amniotic fluid. So in all these cases, we see that these are just before or just after the birth. While the birth of a child is happening, if you are uh, drawing the stem cell from one of these sources, then in that case it is called as perinatal stem cell. Now perinatal stem cell is also very close to adult stem cell, because they also already have grown. It's not that the child has not grown completely. So that's why there is a limitation in their ability to specialize. Now, based on these origins, we see that there are three types of cells called as totipotent cells, pluripotent cells, and multipotent cells. So just try to understand what is the meaning. So totipotent, in a manner you can say that this is being derived from the word total. So that's why totipotent, derived from total. So we see that in this, but in these kind of cases, we are looking for, or we are looking into some of the cells which have an ability to specialize into anything. That's why the word total. So totipotent cells are derived from the embryonic cells during or up to the first two cycles of cell division only. So just after fertilization, within the first two cycles of cell division, it is called as totipotent. All right, just first two cycles of cell division, it is called as totipotent, and after that there will be no totipotent. So after the first two cycles of cell division, after the embryonic development, you don't have totipotent anymore. So this will have an ability to specialize into all cell types. They can be specialized into all cell types in the body, so every cell in the body they can specialize into. Then we have the pluripotent cells. Now, pluripotent stem cell, so these are embryonic. Even pluripotent stem cells are embryonic, but after the first two cycles of cell division. All right. So these are after first two cycles of cell division. All right. So here, in case of totipotent, this is up to the first two cycles of cell division. All right, so this is basically what we will see with totipotent, and after the first two cycles of cell division, the embryonic stem cell will be called as pluripotent cell. Now pluripotent cells, you can say in a manner that they can specialize into almost, let me just add here, almost all cell types. Okay, they can specialize into almost all cell types. So they cannot do all, that was the case with totipotent, but here almost all right, so that is why pluripotent. And then we have multipotent cells. So multipotent cells basically are the adult stem cells and the perinatal stem cells, which have a limited, their ability to specialize is more limited. So because they are found in the adults, so that's why they have a limited capacity to specialize. So this will be found in the adult and the perinatal stem cells, where already the growth into an individual has already happened. So these are called as multipotent stem cells. So this is how we define the different types of stem cells. Now, when it comes to the use of stem cells, we find that there are, of course, there are various different ways in which we might be able to use the stem cells. So, for example, we can use them for something called as regenerative medicine. So, when we say that something is regenerative medicine, the meaning would be that they can be used to regenerate the damaged tissues or organs. So, why exactly? Because we know that the stem cells can grow into new cell types, correct? They can grow into new tissues altogether. So that's why we say that this is regenerative medicine. That whenever you have any injured part of body, some kind of accident, or even due to some kind of, let's say by due to heart attack also, sometimes we see that the heart muscles also get damaged. So even those kind of situations can be cured with the help of stem cells. That stem cells will be used to grow a fully grown tissue out of these kind of scenarios. So that's why we see that this actually can be done with the help of stem cells, and this is called as regenerative medicine, because we are trying to regenerate a damaged tissue with the help, or organs with the help of stem cells. So regenerate damaged tissues or organs. Then after that, in drug testing also they can be used. So when we say drug testing means what? That they can be used to create tissues that mimic the human organs, allowing the researchers to test the safety and efficacy of a new drug without using animal or human subjects. And this has become very, very important of late in the drug testing arena. That what we see is that instead of using a human subject, instead of using animals for testing out new drugs or new vaccines, we instead, what we do is that we take stem cells, we grow tissues or organs out of these stem cells, and then test the medicine, or we test the vaccines on this, so that we understand the positive and the negative impact that this medicine or this drug might have on the humans, might have on the stem cell. So that's why in drug testing also, for a safer drug testing, we see that stem cells can actually become very, very useful. Then after that, they can also be used for disease modeling. And especially when it comes to the development of genetic diseases, we see that in those kind of cases we might be able to use this kind of we might be able to use the stem cells for the modeling of these diseases. So that's why we see that we can create the models, and this will allow the researchers to understand the various steps of development, or the mechanism of the growth and development of any given disease. So that's why that is called as disease modeling. Then there is stem cell therapy. In case of stem cell therapy, we know that there are multiple different diseases that can be uh, treated with the help of stem cell therapy. And stem cell therapy especially has become important in those kind of cases which have been unsolvable for us. They have been unsolvable for us with the help of normal medical procedures. The normal medical procedures, wherever they are not usable, whenever you don't have any medicines or vaccines, etc., in all these kind of scenarios, we have experimented and researched extensively, and we have been successful in certain cases. That even cases of leukemia or lymphoma and other blood and immune disorders, we see that stem cell therapy can be utilized. And then they can be used in basic research as well, that many of the different kinds of things, that the biological processes, the growth and the death of cells and tissues, etc., the various steps of development from an undifferentiated to a differentiated cell, all these things can be very easily studied. And that research will also give us a lot of insight into what is happening inside the body. And if there are areas in which we can improve further in the medical field, that will definitely give us a lot of insight into that also. Then it can be used in tissue engineering. It can be used in organ transplant also, that we would be able to generate new tissues or walls or organs, etc. And that's why it is called as tissue engineering. Then in case of neurodegenerative diseases as well, we see that we can make the use of the stem cells. For example, diseases like Parkinson's or Alzheimer's, we see that there are neurons that we can create, and these neurons can replace the damaged neurons which were causing these kind of neurodegenerative diseases. Then even in dental applications also they can be used. They can be used to damage the repair the damaged teeth, or even in regenerating dental pulp. So in all these cases, we see that we can make the use. Even we see that they find their use in autoimmune diseases as well. So even in autoimmune diseases, we see that we can make the use of stem cells. So that's why stem cells, we understand that there are extensive ways in which stem cells can be used, and the world has been doing that. There is a lot of growth that has happened here, a lot of success that we have seen, but I would still say that there is a long way to go. That we still are not where we can actually be with stem cells. So that's why stem cell therapy is still a very nascent segment, and it is just going to grow from here, and we'll see that there are a lot of things that these kind of stem cells will be able to do. Now, one of the problems, there are certain issues also that we have with stem cells. Two basic issues that we need to understand. First, the ethical issues in the use of stem cells. That if we are using stem cells that we have derived from the embryos, then of course there is an ethical issue, that you would be deriving all these cells, and then during the course of research or study, they may not be able to survive, right? They'll not be able to survive, and hence in these kind of cases, using embryonic stem cells, especially, is something that is kind of not allowed. That's why very easily. But on the other hand, there is a problem with the help in adult stem cells also, because adult stem cells have a limited ability to specialize, correct? They have a limited ability to specialize, so that's why you can't use them as easily or as extensively as you could have used embryonic stem cell. So on one hand you have these ethical issues, and on the other hand you have some technical glitches. So that's why in both these cases we need to find a balance, and there is a balance that we have here, which is called as induced pluripotent cells. Induced pluripotent stem cells. So when we say that something is induced pluripotent stem cell, the meaning is that here we are inducing the properties of pluripotent cells in multipotent cells, meaning that this is where we are trying to convert. So this is conversion of adult into adult into embryonic stem cell. All right. So conversion of adult into embryonic stem cell is called as induced pluripotent stem cell. So that's why in these cases we see that these two issues that we have, there is a middle ground for these two issues, and that is induced pluripotent stem cells, where we take the adult stem cell and we try to convert it into embryonic stem cell. So then in that case it is called as induced pluripotent stem cell. Look at this question. This question was asked in your paper. I'm launching the poll also for you. Take a minute. Please mark your answer. All right. So as I see, 68% of you have given the correct answer. That is wonderful. Look at the answers now. So with reference to stem cells, frequently in the news, which of the following statements is or are correct? First statement is stem cells can be derived only from mammals. Now, whenever you see the word only, it's an extreme word, as I told you. And in most of the cases, these kind of statements will be wrong, correct? And this is the case here as well, that not only mammals, but rather most of the higher plants and animals, most of the higher plants and animals, they basically, most of the higher plants and animals, they have stem cells. So it's not only animals or not only mammals, but rather even in plants there are stem cells. Where exactly? On the tips. All right. So in case of plants, the tips of the plants, the tips of the barks of the tree or leaves, flowers, they basically, they have stem cells, because those are the areas that grow, correct? Those are the areas that grow. So any area that grows, basically they have the stem cells. So basically if they are called as meristems or meristematic tissues, etc. They basically have the stem cell. So in these scenarios, we see that stem cells actually are present not only in animals but also in plants. So that's why the first statement is wrong. Second is stem cells can be used for screening new drugs. We just discussed this, that instead of using an animal or a human subject, we can use stem cells and test the new drugs and vaccines on the stem cells. Stem cells can be used for medical therapies. Of course, stem cell therapy has become so popular over the uh, last decade, you can say. So that's why second and the third statement, both of these statements are correct. So correct answer will be 2 and 3. Now moving on to the next topic. The next topic for us is DNA fingerprinting. Now in case of DNA fingerprinting, this is basically something that refers to identification of partial or complete set of DNA in a given sample. Now overall, when it comes to the word when it comes to the word fingerprint, where do we exactly use the word fingerprint? I forgot to add, if you have any question from stem cells, is there any question that you have that you want to ask, or is it all clear? Either I'm teaching very well or I'm not teaching well at all. Those are the only two conditions when a student does not have a doubt. How much of syllabus has been completed till now? We have completed space technology, the complete chapter. We have completed energy, the complete chapter. And right now we are on the verge of completion. Maybe it will take another 45 minutes to 1 hour to complete this third chapter, that is biotechnology. So these are the three chapters which are done, and then after that the next chapter, probably the last chapter of the day, will be nanotechnology in today's class. And then after that all the rest of the chapters will do in the next week. Pluripotent. So pluripotent basically means that there are multiple cells into which they can specialize. I mean, the stem cells can specialize. So in a manner, you can say that this is being derived from the word plural. So plural means what? That there are multiple different things that are called as plural. So here, pluripotent is when there are number of cells that can be derived from this stem cell. Various different types of cells can be derived from this cell, but not all. Not all. That is totipotent derived from total. So that's the very basic difference. But in both these cases, they are being derived from embryonic stem cell. Short terms are important. See, terms, especially in biotechnology, are important. That's why so many of these technical terms that we have encountered in this chapter, they can actually become very, very important for your examination, because there are huge chances that they will ask you questions from all these areas. All right, there are chances that they'll ask you questions from these areas. So that's why you have to be prepared that these kind of things can come in your paper. Where will I get? I mean, we are discussing it in the chapter, so you will get everything in these PPTs only. I'm asking how much syllabus of this subject is already over. That's what I'm saying. Space technology, energy. These two chapters are over, and the third chapter that we are going on with is biotechnology. All right. So let's move on. So we are talking about DNA fingerprint. Now, when it comes to DNA fingerprint, what do you know fingerprint for? What is fingerprint known for? What is the feature of any fingerprint? Uniqueness, right? Correct. So differentiation from others, right? So that is basically what we are looking at. Correct. That when it comes to uh, fingerprint simply is unique. I mean, my fingerprint is different from yours, and everybody has different fingerprints. Same actually goes for DNA as well. That because especially when you are talking about the higher plants and animals which are born out of sexual reproduction, their DNA also will be different, right? There also there is mixing of DNA, so that's why each and every DNA set is unique. So that's why it is called as DNA fingerprint. All right, that's why DNA fingerprint, and that's why the samples of DNA when we take from individuals, if they are blood related, there will be a lot of similarity between them, and if they are not, then there will be certain differences between them. So that is called as DNA fingerprint, and that's why it finds its applications in a lot of areas. So for example, in identification of criminals, we can find the use of DNA fingerprint. So identification of criminals, so how exactly in identification of criminals, we basically see that, let's say there is a crime scene, and in the crime scene there are a set of DNA that we have found. In the crime scene we have found a set of DNA. So now let's say there are three suspects. So for these three suspects will take the sample of the DNA from them, and they will be matched with the crime sample DNA. So if you see the crime sample DNA, and out of these three suspects, suspect 1, 2, and 3, can you tell me which DNA suspect matches, DNA sample matches? Who is the most potential suspect in this particular crime? I'm waiting for more answers. Couple of you have given the answers. Others are sitting. You can give the answer. Yes. So, as most of you have written here, it's correct. Sample number 2, correct. In sample number 2, you can see that there is a match that happens between the sample that we got from the crime scene and suspect number 2. So at least we would know that suspect number 2 was present at the crime scene. Whether that suspect had done the crime or not, we can't say that, but at least we understand that there is a presence. So that's why identification of criminals can be done with the help of DNA fingerprint. Then claim over dead bodies or claim or paternity tests can also be done with the help of DNA fingerprint, because there will be certain uh, similarities between the DNA of people who are related by blood. So that will also help us in identification of people who are related. Similarly, in making effective drugs, if you remember, there is something called as, we discussed, pharmacogenomics. Correct. We discussed about pharmacogenomics. So pharmacogenomics basically talked about how pharma can be, or how a medicine can be made according to the genetic structure of an individual. Correct. According to the genetic structure of an individual, so that the drugs are as effective as they can be. So that is basically that is something which is called as pharmacogenomics. So to make effective drugs as well, we see that we can make the use of pharmacogenomics. Then treatment of genetic vulnerability. Now, in case of genetic diseases as well, we see that we can make the use of genetic testing. If you remember, we discussed about this as well, that genetic testing many a times can help us in identification of something. So in case of genetic disease or any disease where an onset is possible in the future, we might be able to get a hint about this onset from from the testing of the genes. So in these cases also we see that testing, genetic testing, can be very, very helpful. Overall, that's why it becomes very, very important. Presence or absence of certain genes will give us an idea about the future onset of disease. So that's why we see that all these cases. So I'm writing it out here. Presence or presence or absence of certain genes. Presence or absence of certain genes can hint towards future possibilities of a disease. And then finally for wildlife management as well, that in case of wildlife management also we can actually make the use of DNA fingerprint, because in wildlife management the thing about wildlife management would be that we want to understand what are the external factors, what are the anthropogenic factors, or what are the climatic or environmental factors that are, let's say, leading to the decrease in population of a certain species. So if we are able to find that, if we are able to find that particular event properly, then it will give us an idea about whether or not there are some things that can be done, and all the conservation practices and all the steps that we want to take to conserve these particular species, these all things can be done with the help of understanding of DNA fingerprint. So that's why DNA fingerprint can be very, very useful. At the same time, we also want to preserve the genes of something which is critically endangered. Any species that is critically endangered, we would want to save them as well. So that also can be done with the help of DNA fingerprint. So that's why there are multiple advantages that we have, multiple applications, but at the same time there are issues also, that concerns also that we have over DNA fingerprinting. And what are these concerns? Also understand this, that it can lead to certain issues. So what are the issues? First, that information can lead to reference for designer babies. Reference for designer babies. If you remember, we discussed about the fact that we can pick and choose genes. Now, with reference to when it comes to DNA fingerprint is all about identification of certain genes, similar, dissimilar properties, whatever it is. So that's why when it comes to the reference for designer babies, this is also where we might find that there is a chance that they might be, we might identify certain genes and we might want those genes in the children. So that's why this can become very harmful trend altogether. And this is where there is one term which is called as germline editing. There is this term called as germline editing. So this germline editing is something that somebody might try. Germline editing is when we make the changes. All right, this is making permanent changes. Permanent changes in the genetic makeup of an individual. So especially when the changes are done in the reproductive cells itself, then these will carry on from generations to generation. So okay, let's say you want that your child should be a great mathematician. So then in that case, you will try to pick the genes of somebody who is wonderful in mathematics, maybe one of the greatest minds in mathematics right now. And you would pick those genes which are specially responsible for those kind of cognitive abilities. And you would want that this particular gene must be there inside this child. And what you might do is that you do this change in the reproductive cell itself, so that this, when it translates, it actually becomes a permanent change in the next generation as well, and it can carry on from those. Now you might think that you have done a great job, that you have produced a wonderful mathematician as your child, but there is something called as a consent. This is where this is actually an ethical issue, that you have taken the decision for the child, whether the child wanted to be a mathematician or a historian. You have no clue about it, you don't care about it, you just have taken the decision that you wanted to take for the child. So that's why it is completely unethical to do something like this. Although germline editing, it's not that germline editing is completely banned. Germline editing also can be good in treating genetic diseases. So if you, let's say there is somebody who has a genetic disease, we can make a germline editing in the genetic disease, in treating that genetic disease, so that the genetic disease can be completely removed, and for the future generations also that genetic disease can be removed. So in that case, that scenario, germline editing is good. But in other cases where we are now looking at certain scenarios like designer babies, etc., this can become a very dangerous trend going forward. Then genetic privacy of individuals will be violated. That also is possible, that let's say if the genetic information about individuals is available publicly, then everybody knows what are so-called good genes and bad genes. So you might feel that, and if you know, you might see that, okay, this person has bad genes, so now this is new kind of classism, right? We find new ways to be classists, and this probably will become one of the other ways to do that. So obviously, look at the world and you know that there is racism, there is casteism, uh, there are so many ways in which we differentiate between people on the basis of whatever it is, whether it is language or religion or caste or skin color or whatever it is, ethnicity. I mean, the list goes on in this world. So this will become a new way for us to become again become classist. That will see that, okay, fine, I have good genes, so I am better, I am superior, and this person has those bad genes, so that person is not in the same pedestal as I am. So that becomes another new way for us to start differentiating between people. Then at the same time, genetic information may be misused for commercial purposes as well. And we might see that medical companies, pharmaceutical companies might start offering you packages that, look, you have these genes can lead to these kind of complexities for you in the future. So this is what we offer you. This is the medical package. If you take this, all these future possibilities of all these diseases can go away if you take this package. Now we don't know how they are be using it. They might be correct, they may be completely wrong. It's just that they are trying to sell what they understand, sell the information that they have. So, and that is a very dangerous trend. And forget about what they can do in the future. Even today, uh, you can't deny that science is not misused against you many a times. Correct? You, let's say you go on to buy cosmetics or let's say electronics, you'll find that there are so many terms that are written, that this has this has that, this is that, this will have this effect and that will have that effect. They will keep telling you all this about, and in most of the cases you have no clue. You have no clue that whatever is being sold, why exactly is it being sold. That, for example, charcoal. All right, so this is a trend that we have today, that you have charcoal in your face wash, charcoal in your soap, charcoal in your toothpaste, charcoal in your face wash, charcoal in everything. So you might think, oh fine, charcoal must be good, that if there is an expensive product that I'm buying that has charcoal, must be some reason, must be the case that I'm buying, I've been asked to buy this. So unfortunately, many a times we don't have an idea about what exactly charcoal does, or whether charcoal is really required. Then why not just go and buy some coal, buy some charcoal and start eating it if it's so good for you? So we don't know how much. We just know that the things will be very well advertised to ensure that you fall into that trap. So unfortunately, science can be misused because of lack of knowledge at our end. Lack of knowledge at our end, we do not understand, and that can become the criteria. There's a wonderful book written on this particular subject. The name of the book is Bad Science. So this book, Bad Science, is all about all these practices, that how various industries, how various sectors will use the misinformation that you have about science or about technology. And that's why they would sell you anything, just that they have to be believable. They don't have to be correct, they have to be believable. And if they are believable, you will believe them and you probably will buy the product, just because they told you about a lot of things that sounded very fancy and sounded very believable, and they made you believe that this is happening, this is correct. In this book, Bad Science, this author also mentions an incident about his own life. that when a certain company had come to him and they said that we are going to publish a research, a fake research in the newspapers, we just want to use your name. We just want to use your name, and if you give us the consent to use your name, we'll pay you X amount. This much money will be paid to you if you just let us use your name for this fake research. The author refused. The author said that no, this is completely unethical, I'm not going to do it. So he refused the offer. 1 month later, he says that one fine day he was reading the newspaper, and then he just studies about certain research and sees that that's the exact same research that the company was talking about, now published with some others name. So ultimately what happened is somebody accepted the offer and they let their name be published, uh, and a fake research was published in the newspaper. Now what does this fake research do? It talks about certain problems. It talks about certain issues, that this study has found this problem, that study has found that problem, and 2 or 3 months later they release a product, a cosmetic product in the market which takes care of that issue. So first, a fake paper, a fake research has been published. You have read it. You now know about the issue. Now, 2 months later, a new product is being advertised, which is telling you that these kind of issues, which are recently being reported in many researches, now we have the ability to solve it. And out of your own insecurities, you go and buy that product. So this is how many of the products sell. That first you create that fear in the minds of the consumers, and then dwell upon that fear, and then sell your product based on that fear. This is how sales a lot of times work, especially in the electronics industries, or especially forget about electronics, at least in healthcare, nutrition, etc. It is where this is how it sells on your insecurities, your fears. So we know that these things can actually become a reality. Then genetic information may be used to even create weapons of mass destruction. Correct? This also can be done. That we might see that there are chances that people start creating weapons of mass destruction based on that. How exactly can they do that? They know about pathogens, they know about the genetic makeup of those pathogens, and that can actually be used in making anything which is weapon of mass destruction. So, for example, let's say uh, they can regenerate an extinct pathogen. So if you consider, let's say, smallpox, all right. So smallpox now has been eradicated because wide vaccination had happened all across the world. But this is now 3, 4 generations old. So the people who are born now, they might not have a natural immunity against smallpox. So if smallpox today enters the world again, then people will suffer from it. People will be affected. So that's why these kind of situations can actually happen, and this is also a concern with DNA fingerprint. So that's why there are so many problems that we have when it comes to DNA fingerprint, and we really need to be aware of all these problems so that we know how to solve these problems when time comes. Then after this we have gene therapy. Now we discussed about gene therapy. We discussed how exactly either replacing a gene responsible for responsible for disease with a healthy gene, or inactivating or knocking out a gene that is functioning improperly. That is also something that we can do, or introducing a new gene into the body to fight a disease. So either, as we had discussed, addition, subtraction, replacement, correct? Addition, subtraction, replacement. So similarly here also the same three steps we are talking about. Now when it comes to gene therapy, there is, although we have seen that it has shown a lot of promise, and we have been implementing gene therapy in kinds of cases where the problems seem to be unsolvable. We have tried to solve those issues with the help of gene therapy, but at the same time there have been certain concerns over gene therapy as well. First concern, for example, has been the short lived nature of gene therapy. That we see that gene therapy does not live for very long, and the reason for that many a times has been the fact that gene therapy gene therapy, when introduced into the body, it starts working, But in our body, you might know that the cells always keep on dividing. Cells also keep on getting replaced with new cells, and this process keeps on happening very quickly. I mean, by the time I finish just one sentence, almost 10 lakh cells in my body would be replaced. So such is the pace of replacement of the cells. So cells will be born, cells will be functional, cells will die, and this will keep on happening all the time in the body. Now, while that happens, many a times the cells, while going through this entire process, might overlook the new development or the new introduction of a new gene, and might go back to the original ways, and hence it can lead to the gene therapy having a less may have a short term uh, effect on the body. So that's why this is one possibility. Second is about the immune response, that many a times what happens is that our immune system itself causes the problem, that the immune system itself uh, will not let the gene function, because immune system lives by a very simple philosophy: anything that is foreign is my enemy. This is how immune system functions, that it does not recognize what is good gene, what is bad gene, what is helpful, what is dangerous. It just understands one thing and one thing only, that anything that has come from outside, anything that is foreign, will be dangerous for the body, and hence it must be removed, hence we must get rid of it. And that's why the body, or rather the immune system, will try to get rid of it. So many a times the immune system itself might attack the gene that is not recognized well by the immune system, and that too can lead to gene therapy not working well. Then there is one more problem, that there are certain diseases which are multi-gene disorders. All right, these are multi-gene disorders, meaning that it's not only one gene that gets affected, but there are multiple genes that get affected at the same time. And in such scenarios, it's very very difficult for us to replace multiple genes at the same time. It's very difficult, it's very very complex as a procedure. So that's why in these kind of cases, gene therapy is not that useful, not that helpful. So, for example, heart disease, high blood pressure, Alzheimer's, arthritis, diabetes, all these are cases where we see that something like this happens. So, overall, there are these issues, plus also adding to that the cost of the therapy as well. That right now, gene therapy is costly. All right, gene therapy is costly, and it's not that easy for us to uh, for everyone to afford gene therapy. Only people with that kind of capital availability can afford gene therapy. Otherwise, common public can't. So that also is one of the areas where we need to improve. We need to find ways to reduce the cost of gene therapy. We see that there are certain steps being taken by the Ministry of Health as well, so that they can reduce the cost of all these therapies, so that more and more people can at least have uh, a value for this, that they would be able to go for these therapies in times of requirements. One more concept which has been in the news very frequently is the concept of three parents baby. Now the concept of three parents baby basically talks about the fact that there are cases it talks about the fact that there are cases when the mitochondria of the mother cell might be defective. All right, mitochondria of the mother cell might be defective. Now, whenever this happens, that mitochondria of the mother cell are defective, the property of mitochondria is that mitochondria get transferred from the mother to the child, never from the father to the child. All right, never from father to child, always from mother to child. This is what we see with the mitochondria. Now, if the mitochondria in the mother's cell, if they are defective, then in that case, the same defective mitochondria will go to the child, and then the child will also have the same mitochondrial disease that the mother has. So, to solve this problem, this concept of three parents baby basically works or has been introduced. All right, so what we basically see here is that if you look at this, we know that there are unhealthy mitochondria that the mother's egg cell has. So now what we do is that we take a donor, the donor has healthy mitochondria, and then we do a replacement in a manner that these diseased mitochondria will be uh, replaced with healthy mitochondria. So for that, what we do is we again remove the nucleus of the donor cell, and the nucleus from the mother's cell is introduced into the donor cell. Now, if I ask you, mitochondria was defective, why are we replacing the nucleus? Why did not we simply replace the mitochondria in the mother cell instead of going through this indirect process where from the donor cell we are taking away the nucleus and then inserting the nucleus inside the donor cell, and then the fertilization happens between the donor cell and the sperm cell of the male? Why? Why not directly replace mitochondria? Think of an answer. Give me some answers. I see some of you have given the answers, but I'm expecting more. There's no negative marking. So just think what you see as a possible reason that why do we replace nucleus instead of mitochondria. Yes, partially, Himanshu, your answer is correct that because the offspring will bear the characteristics of donor mother, so that's why nucleus definitely has to be gotten rid of. So I'll tell you the reason that why not directly replace the mitochondria and why replace uh, in the donor cell, why replace the nucleus? Because there is only one nucleus. There's only one nucleus and there are multiple mitochondria. So if you start picking up the mitochondria and start replacing them, it is not going to be an easy task. It's going to be a very, very difficult task because there are multiple mitochondria in one given cell. So that's why carrying out this entire process will be too cumbersome. It will be too complex. Secondly, we already know how to do nuclear replacement. We know about the process of cloning. So in process of cloning, we know how to do the replacement of the nucleus. So that's why we can replicate the similar process here as well. So that's why instead of doing a direct replacement in the mother cell itself, we take the donor cell and replace the nucleus there. And why nucleus? Simply because why nucleus? Because nucleus has all the specific characteristics. All right, nucleus has all the specific characteristics. So if you have taken away the nucleus of the donor and you have replaced it with the nucleus of the original mother, then in that case the child will also have all the characteristics of the original mother and the original father, right? So that is why nucleus. So so Binita, yes, your answer was perfectly correct, that it's easy to replace nucleus than mitochondria, and that is basically the main reason why we do something like this. Now there is another question here, that it's not only nucleus that has DNA. We saw that in the difference between DNA and RNA, that not only DNA but also mitochondria also have the DNA. So just remember this. All right, there are these three organelles that have DNA in them: nucleus, mitochondria, and in plants the plastids. All these three, they contain DNA. So it's not only the nucleus that contains DNA, but the DNA that is inside the nucleus is specific to that individual, whereas the DNA that is in mitochondria or plastid is not only uh, individual characteristic in nature, but instead it has the characteristics of the entire species. Meaning that my nuclear DNA will be different from your nuclear DNA, but my mitochondrial DNA and your mitochondrial DNA will be similar. So every human will have a similar mitochondrial DNA, whereas the nuclear DNA in each and every individual will be different. So that's when nucleus becomes very important. And even if mitochondrial DNA is there, the donor mitochondrial DNA is there in the child, it will hardly make any difference in the characteristics of the child. So that's why the child still belongs to the original mother and the father. There has been a controversy here, that there was a case that was actually fought in the UK about this as well, that should the donor should the donor also have a claim over the child in such scenario? And this was the court ruling then. The court also simply said that no, only the original mother and the original father will have the claim over the child, and the donor will not, because donor could have been replaced with thousands of other donors which would who would have the similar mitochondria, but that cannot be done for nucleus. So that's why the child, I mean, whoever the nucleus belongs to, whoever the nuclear DNA belongs to, they will be the people who will have the claim over the child. So already these kind of controversies have actually happened here. There is this question also that was asked in your paper, that in the context of recent advancements in human reproductive technology, pro nuclear transfer, sorry that this somehow got shifted, pro nuclear transfer is used. So pro nuclear transfer is actually another name for the same procedure. So we also call it as 3% in vitro fertilization, mitochondrial replacement therapy, pro nuclear transfer technology, three parents baby concept. There are multiple different ways that names that we use for this therapy. So basically, this will be for the prevention of mitochondrial disease in the offspring. So that is why we will be using this kind of a process, right? Then after this, one more topic that has been in the news has been GM mustard or Dhara mustard hybrid 11. So this Dhara mustard hybrid 11 is a genetically modified GM mustard. Now in this case, when it comes to DMH 11, DMH 11 was in the news back in 2018-19 also, and again it has come back in the news in 2022. Now, when it comes to when it comes to DMH 11, this DMH 11 has been in the news because of certain controversies also. In 2011, we were about to release the seeds of DMH 11 for the Indian farmers. The Genetic Engineering Appraisal Committee had given its go ahead, and the government of India also had given its go ahead. But then this is where Supreme Court intervened, and Supreme Court did not let the seeds of DMH 11 release into the market, and then it was sent back to a panel to do further studies and further research on DMH 11 and bring back a report about the safety concerns that many had with regards to DMH 11. Last year, 2022, this committee that was set up, it came back with its report, and they said that there is no problem that they have found, and they think that this DMH 11 is ready for the Indian markets, that we can release it for the Indian markets. The final approval from the government of India is still pending, right? It is still pending. We are just still waiting for the government of India to give the final go ahead or the approval. But other than that, the panel, etc., they have already cleared it. So from that perspective, we see that uh, this is basically where we see that DMH 11, it can actually uh, be released if, let's say, given the go ahead, it might be released for the market. Now why exactly DMH 11? Because DMH 11 basically can give up to 25 to 30% better yield as compared to the best varieties of mustard that we have in India, for example Varuna. Now, and this is something that was developed by Center for Genetic Manipulation of Crop Plants in Delhi University. So this happened in Delhi University. So so in Delhi University, this basically was prepared. So the problems that happened with DMH 11 was mainly there are certain issues, some environmental concerns or biodiversity concerns, I would say. There are some concerns related to biodiversity, and there are some concerns related to the human health as well. Biodiversity and human health. So these are two primary problems that we saw that would happen. Why biodiversity? Because right now in India, there are almost 66 different variants of mustard that we grow. And when DMH 11 comes with such better yields, if the tests show the same result as the actual result, then in that case there are chances that many of the farmers who are growing various different types of mustard variety may stop growing those mustard varieties, and we'll see that more and more of mustard varieties will be replaced with this DMH11. So that will have a long term cascading effect, that slowly and steadily many of these mustard variants might start becoming extinct. That also becomes a possibility. So that is the reason why we see that there are all these issues that were raised against DMH11. And this question, that's why it was asked in your 2019 paper. Just look at this question, read the question, and I'll launch the poll for you. Yeah, so I see very mixed answers, that 39% of you have given B, 22% C, and 33% D. This was a very, very easy question. This was a very, very easy question. Look at statement number three. I'll give you the answer from here. Look at statement number three. GM mustard has been developed jointly by IARI and Punjab Agricultural University. What did we discuss? That it was this was developed at Delhi University. There is no Punjab University here. This is wrong. Statement number three is wrong. You have three here, you have three here, you have three here. The answer is B, two only. So you directly get the answer from here. So from one statement, and where we discussed that this was developed at Delhi University. So that's where three is wrong. So that's why we are left with anyway just one statement, that is statement number two. So statement number one says that GM mustard has genes of a soil bacterium that gives the plant property of pest resistant to a wide variety of pests. That's not correct, because this is what is close to what BT cotton is. So that's why it's not about this mustard, because here we are not really talking about the resistant to wide variety of pests, but rather to increase the productivity. So this is wrong. The answer is to increase the productivity. So that's why one can't be correct. All right. So correct answer will be 2 only. Now, coming to the last topic of discussion for biotechnology, and this is dark biotechnology. Now, in terms of dark biotechnology, what we basically see is that dark biotechnology denotes the misuse of biotechnology. When biotechnology is being misused or being used for wrong purposes, it is called as dark biotechnology. And there are various ways in which dark biotechnology comes into the picture. The first one is bioterrorism and the use of bioweapons. That in case of bioterrorism is any act of violence, whether it is against humans, against human lives, against our crops, against our livestock. It could be anything. Harming even the environment of a country deliberately will also be called as an act of bioterrorism. So that's why bioterrorism can become a reality with genetic engineering, with better understanding of genes, we might see that something like this may happen. And then after that, the use of bioweapons, that when we discussed in DNA fingerprint as well, that there are chances that we'll be able to regenerate a given pathogen or make a new pathogen in the laboratory. For example, let's say if you, even if you consider COVID, is it not that there are many theories that say that COVID-19 virus was developed in a lab in China? Some even Chinese scientists have said this. US report says this. So there are some people who say that this actually might have happened, that they produced this COVID-19 virus in a lab. So we would never know whether that's correct or wrong, because you will never get a transparent information out of China. But at least we know that these are some of the discussions that have been happening with regards to the biological weapon that perhaps this COVID-19 would have been. So regeneration of extinct pathogens, we had discussed that even this is a possibility, that we might be able to regenerate extinct pathogens, and even that can have a very negative impact on human health and even environmental health. Then effects on biodiversity. Now, when we look at the effects on biodiversity, what we understand is that when it comes to biotechnology, biotechnology may have a negative impact on may have a negative impact on biodiversity. How exactly is that true? Because when we study biotechnology or whatever applications of biotechnology we see, we often are concerned only about certain species, right? We are not making an effort for each and every species of the world. There are certain species which are so called useful for us, and we only are taking all the steps possible for these useful species and not going beyond that. So this is a scenario where we see that it has a negative impact on biodiversity, that it might lead to decrease in biodiversity because we are only concerned about a few species, um, uh, instead of having uh, focus on all the other species as well. So that's why this is one problem that we have with regards to effect on biodiversity. Then there is a problem called as terminator genes. Now, in case of terminator genes, what we see is that this is where there are genes which sometimes are inserted in the seed in a manner that the seed will germinate only once. All right, the seed will germinate only once, meaning that let's say you go to the market and you buy some seeds of one particular plant, and you come back and you plant those seeds, and you basically get, let's say, a very good productivity, very good quality and quantity. So then what would you want? You would want that you conserve those seeds and also use it in the next season as well, because you have seen a very good produce. Next season, when you use the same seeds from the fruits or the vegetables that had grown, you see that none of these seeds germinate. These seeds will not germinate because the seed company would have had added terminator genes in these seeds. So that's why when terminator genes are added, we see that the terminator gene will stop the plant from releasing fertile seeds. So this is where the problem is, that when it comes to terminator genes, the terminator genes will basically, whenever terminator genes are used, the seeds will germinate only once. The seeds will germinate only once. So in the next generation, the seeds from the same plants cannot actually be used again. So that is where the problem lies when it comes to terminator genes. So one thing is fine. We understand that this will have a negative impact on the farmer's economy as well, that farmer also will have to just go back all the time and buy the seeds. Now what happens is that last year the farmer has seen a good, very, very good produce. So maybe they want the same seed again and they go back to the market, and this time we see that uh, they would see that the prices would have increased, that seed prices would have increased, that ultimately, because this is how the profits will be gained by all these seed companies. And many of the biggest seed manufacturers in the world, um, uh, they have been, at one point or the other, they have been accused that they are in this unethical practice sometimes. Then what may also happen is that the same may also cross pollinate with local varieties and may affect the continuity of agriculture. That what may happen is one thing, right? What we understand is that okay, fine, there is a terminator gene that we had in this plant seeds, so that's why they will not germinate in the next season from the same seeds. At the same time, what may also happen is that these two plants may also cross pollinate, because many a times in a farm it's not only one kind of crop that is grown, the farmer may be growing multiple crops, correct? So if the farmer is growing multiple crops, then it is possible that one crop cross pollinates with another, and the terminator genes are passed on from one crop to another. So then it's not only that only one crop will not germinate, the other crop also being grown here will also not germinate. So this is also one of the biggest challenges that we have with regards to terminator genes. So overall, this is basically what we see as some of the issues that we have with dark biotechnology. That there are multiple ways in which we might misuse, biotechnology, and uh, that might happen in certain cases. Now, this basically brings us to the end of this particular chapter. Let's take a break here. But before that, if you have any questions with regards to whatever we have discussed, you can let me know. Mitochondrial DNA proves the out of Africa theory of early humans. Yes, that is actually correct. That mitochondrial disease actually is a big evidence for the correctness of the out of Africa theory. There is a wonderful paper also that was published in the Science Journal about this. So that's correct. If a DNA test of the child is done, how much chance of match will be there with the donor? There will be no chance, because when we do the DNA analysis, when we do the DNA testing, we actually do the DNA testing of the nuclear DNA, not the mitochondrial DNA. So that's why there will be no DNA matchup that will show up with the donor. What is the nerve cell that Soviet used to poison people? There were several, but one was sarin. If you know about this, there was one which is called as sarin. Then there is another one which is called as tabun, and then there is another one which is called as VX. I think there is one more which is Soman, if I'm not wrong. So there are all these different types of nerve agents that were actually used and have been used not only by Soviets but also in many a times in wars they have been used. But these are not biological, all right? These are not biological. These are all chemical weapons. They will be counted as chemical weapons and not biological weapons. All right. So yeah. So I think these were some of the questions that I had here. So let's take a break. Right now the time is almost 5:15. So let's come back by 5:45 and we'll discuss our last chapter for the day, that is nanotechnology. So we'll discuss nanotechnology and we'll end the day with nanotechnology.